Sylvester, you've been gone to cell therapy for a long time. While you've been away, so many things have happened to me. I went through prophase, metaphase, anaphase, and telophase. Now we have Sylvia, our daughter cell.
Don't tell me you split. Why have you done this? And without consulting me?
Well, Sylvester, I've done this for the common good. This was meant to happen. It will create less work for the both of us. This aided in the growth, maintenance, and repair of the body.
Oh, Celeste, I love you. I can't believe you went through an entire cycle for me. Look, she even has your nose. And well, everything else for that matter. You guys look identical.
Here, let me show you what I went through. This is what I looked like when you left. I was still in the process of interphase. I had just gone through S phase, where my DNA replicated to form sister chromatids, joined together by centromeres. Oh, and look, this is my G2 phase. I was so tired after I replicated my DNA, so at this stage, I rebuilt my energy reserves. I also manufactured proteins and other molecules to make structures required to divide my nucleus. But mitosis is where the fun really started. During prophase, my chromatin condensed into tightly packed chromosomes. My nuclear membrane degenerated, and my chromosomes mixed with the cytoplasm. My centrioles moved to opposite poles, creating the spindle apparatus. This network of spindle fibers helps chromosomes to move within me. During metaphase, 2 spindle fibers from each pole attached to my centromeres and guided my chromosomes to my equatorial plate. They attached in such a way that my sister chromatids were facing the 2 opposite poles. In anaphase, all of my centromeres split apart and sister chromatids are separated. My spindle fibers shorten and my sister chromatids are pulled to opposite poles. Other spindle fibers lengthened and began to push the poles in different directions. The same number and type of chromosomes are found at each pole. Telophase was last. Once the chromosomes reached the poles, they unwound into chromatin and became less visible. The spindle fibers broke down and a nuclear membrane formed around each set of chromosomes, creating 2 nucleuses. Telophase ends with cytokinesis, where all of the cytoplasm splits into 2 cells from an indentation in the equatorial plate. Now Sylvia and I are 2 new daughter cells, and we are in G1. Let's go through it in full speed.
Thank you for everything. You really are the most efficient unit of life on Earth.
Sylvester, where are you? Ah! What have you done to yourself?
Sylvester is no more. He has been replaced by the haploid gang.
I'm Remy.
I'm Jojo. I'm Kai.
I'm Poe.
No! Don't tell me he went through meiosis.
Yes, he split. Twice.
But he was a germ cell.
It's what he's meant to do.
He's gone?
No, he lives on in us. Let us show you how.
There's Sylvester. That's when he was in interphase, and his DNA had just been replicated. It's his S phase.
Yes, and after his G2 phase, he can then start meiosis. Just remember, the chromosomes with dark tips are from Daddy, and the plain ones are from Mommy. First is prophase 1. This is when the nuclear membrane degenerates. Then the centriole splits, and parts move to opposite poles, creating spindle fibers in the process. At this point, we are able to see tetrads.
Oh yeah, tetrads are composed of 4 sister chromatids from a homologous pair. Now if you look closely, you can see the chromatids from the homologous pair crossing over. During this event, DNA is exchanged, creating a greater genetic diversity. During metaphase 1, a spindle fiber attaches to the centromere of each chromosome. A spindle fiber from one pole attaches to one pair of sister chromatids, while a spindle fiber from the other poles attach to the other pair of sister chromatids. The spindle fibers guide the chromosomes to the equator so that the homologous pairs are on top of each other.
During anaphase 1, spindle fibers shorten and pull the homologous chromosomes to separate poles of the cell. The whole chromosome. This is called segregation. In telophase 1, cytokinesis occurs, splitting the cell cytoplasm. Chromosomes uncoil and the nuclear membrane is formed. These cells are now all haploids.
Now meiosis 2 begins. It's important to note that the chromosomes don't replicate before meiosis 2. In prophase 2, the nuclear membrane dissolves and spindle fibers form.
In metaphase 2, the chromosomes, each with 2 chromatids, are guided to the equilateral plate. Then, in anaphase 2, centromeres split and the chromosomes migrate to opposite poles.
Telophase 2 is last. The second nuclear division is completed and cytokinesis occurs for the last time.
Now, full speed!
So now you see, our chromosomes are all different, but we are all part of Sylvester. Now, we are ready to meet with other gametes so that we can continue the creation of life.